Welcome. In this video, I'll be covering the topic of oxidative phosphorylation and the electron transport chain. So what is oxidative phosphorylation? Well, essentially, we're using enzymes to oxidize molecules, in this case glucose. We're stripping them of their electrons, using oxygen as the final electron acceptor. And this is different than substrate level phosphorylation, where you take the phosphate group and just directly transfer it between carriers over to make ATP. Now, while substrate level phosphorylation provides a quick source of ATP, it's not nearly as efficient as oxidative phosphorylation. Because of this process, aerobic organisms, they can produce between an estimated 30 and 38 ATP per glucose molecule. For comparison, fermentation, which follows glycolysis to keep it running, it only produces a net of 2 ATP per molecule of glucose. You can see how much more efficient this is to use oxygen. So remember, prior to this step here that we're talking about today, glucose must be lysed through glycolysis, right? And during glycolysis, you cleave this 6-carbon molecule into 2 3-carbon molecules known as pyruvate, which can then enter the mitochondria and go into the Krebs cycle, right? And so they meet up with their coenzyme becoming acetyl-CoA and through the citric acid cycle, creating NADHs and FADH2s and a couple ATP. So what's going on here is these molecules, NADH and FADH2, are what are known as electron carriers. So they are going to take the electrons from this process and carry them over to the next step, which we're going to talk about in a second. So far, for one glucose, all we've gotten is 10 NADHs, 2 FADH2s, 10 hydrogen ions, 2 ATPs, 2 GTPs, 6 CO2s. So only 2 ATPs for this whole process. So how do we get the remaining 28 or more ATPs from this glucose? That's where we make our money in this next step. We take these NADH and these FADH carriers, we transfer the electrons over to this membrane right here. This is the inner mitochondrial membrane. Okay, this is the outer membrane of the mitochondrion, right? In the inner, I'm going to zoom in here. This is the membrane that I'm talking about. This is what's called the electron transport chain right here. So it's called that because it's literally a chain of these 4 main intermembrane proteins. They're embedded within this membrane. And these complexes, they're scattered throughout the mitochondrial matrix. In fact, this membrane here is highly invaginated. And why? To create more surface area to allow you to put more of these into that membrane. So this powerhouse of the cell is loaded with these things and can create lots and lots of ATP, right? So what this looks like, this series of these 4 proteins, the first one here at the highest energy state, this first protein, accepts electrons and then passes them down this chain, down their potential energy state to this lowest state before those electrons get passed on finally to oxygen. So oxygen really, really wants to grab onto these electrons, right? It's very electrostatic. So, but we're going to take some of that energy and slowly release it as we pass it through here so that we can use that potential energy to do work for us. What is the work we're trying to do? Well, in this case, we're going to drive the pumping of hydrogen ions across this membrane, right? So we're doing this work because anytime you pump something against its concentration gradient, we're making a higher concentration over here. That takes energy to do that. The energy is coming from the NADHs and the FADH2s, right? So we're going to create what's called an electrochemical gradient up here. It's electro because these are hydrogens ions, which are positively charged. So we're creating a higher positive net charge over here compared to on this side of the membrane, right? Because we have so many of these over here. And so that creates an electrical gradient. It also creates a chemical gradient, or a concentration of these ions on the one side. Remember, everything wants to fall down its potential, its concentration gradient, right? So this potential created can be used to do more work. So we're going to trade this energy, run it down here, and create a high potential on this side. Now I should mention that another part of this is when this oxygen grabs onto these electrons, it also grabs onto some of these free hydrogen ions. And of course, that creates water. But what that means is that we're creating an extra bit of potential difference between these two sides, because now you've sucked up some of the positive charge on this side with the oxygen, creating more positive charge on this side. So an even more of a potential difference between these two sides. So these want to get across even more. So now that these want to fall back down, we use that, we let them fall through this complex right here. This is called ATP synthase. This is where we make our money right here. So this thing is going to generate those extra 28 to 36 or so of those ATPs per glucose molecule. And so what this thing looks like is actually something more like this. So now we've kind of flipped that upside down from what you were looking at before. So now this is the inner membrane of the mitochondria, okay? And so this portion right here is kind of like the hub, and it's stuck in the membrane. And then there's this axle that goes through the center of it. So this axle literally rotates around as hydrogen ions pass through this thing. So hydrogen ions pass through, and they go through this hub, and they force this to turn, right? And that turning force, so this axle is actually what they call asymmetrical, because it has a big hump on one side. And that hump, as it spins around, it pushes on this portion, pushes out on these different globular structures here. And each one of these, there's actually, within this portion, there are 3 different pockets, right? Those pockets hold on to ADPs, adenosine diphosphates, and free phosphate groups. So inorganic phosphate sticks into one of these pockets. So there's 3 of them. That bump comes around and changes the conformation of these proteins, forcing those phosphates onto the ADPs, making ATP. So now we've just stored a whole bunch of energy in the form of ATP, and each time this thing rotates, it bumps into 3 of those. So each rotation, 360 degrees, creates 3 ATPs, all driven by those hydrogens that want to flow through back down their potential that we created using electrons that we stole off of originally that glucose molecule. So this is how we put fuel in the form of glucose and get energy out in the form of ATP.